Welcome back to the Duck Dive. So if you're trying to keep up with that constant flood of new scientific papers, new data analyses, just imagine this.
Imagine condensing 6 months of really specialized iterative research.
The kind of work that a PhD student would spend half a year on.
Right. And compressing that into a single 12-hour computational run.
That is just an astonishing acceleration factor, and it's what we're talking about today.
We're looking at an AI scientist system called Cosmos. And it's really changing the timeline of data-driven discovery. The human collaborators who worked with it were very clear.
What did they say?
They said a 20-cycle run did the equivalent of 6 months of their own professional research time.
6 months. That's our starting point. Our mission today is to really get into the mechanics of Cosmos. How does it manage to sustain that kind of deep, long-term focus?
And what's surprising things is it actually finding. We're going to look across metabolomics, material science, even genetics.
And crucially, what does this augmented future really look like for the human scientist in the loop?
Well, this is a huge leap forward because historically, these AI agents designed for open-ended discovery. You might remember systems like Robin or Sakana. They always struggled with coherence.
Coherent?
Yeah, they lose context, they start repeating themselves, or just wander off from the main goal after a few hours. They got tired fast.
So if the last generation of AI scientists got distracted and needed a coffee break after a few hours, Cosmos is the one that can stay up all night working.
That is the perfect analogy. It's broken that barrier.
Yeah.
Its ability to manage these complex, sustained runs is the real innovation here.
And the raw numbers are just staggering.
They are. A single Cosmos run can execute, on average, 42,000 lines of code.
And it's not just coding, it's reading too, right?
It reads over 1,500 full-length scientific papers to find context for its analysis.
That's massive. And I love this one detail the collaborators mention, something that should make every research institution's ears perk up.
Oh, the linear scaling.
Exactly. They found that the number of valuable scientific findings scales linearly with computational effort. You put in more cycles, you get proportionally more science out.
It is the closest thing we've seen to turning abstract research time into a scalable, predictable input.
Okay, let's unpack that. If coherence was the killer for earlier AIs, what is the secret sauce in Cosmos's architecture? What makes it so different?
It really all comes down to its unique architecture, which is centered on a structured world model.
A structured world model. Okay, so that's not just a memory bank, is it?
No, think of it more like a central dynamic library. Or maybe a master digital whiteboard that's constantly being organized and cross-referenced.
So it's not just a chat log of its past thoughts, it's actively structuring the knowledge.
Precisely. Cosmos starts with an open-ended goal like find the cause of fibrosis and a data set. Then it delegates.
To who?
To two specialized parallel agents. One is a pure data analysis agent, it crunches the numbers. The other is a literature search agent, it finds the context in papers.
And this world model sits between them, sort of mediating the conversation.
Yes. If the data agent finds a significant protein, it writes that finding to the world model. The literature agent sees it and immediately runs off to find every paper on that protein, updating the model with that new context.
So that's the key. That's why it can run for 8 times more iterations than older systems.
It's all about that context management strategy. It allows for that deep, sustained focus over 12 hours, doing an average of 200 of these agent rollouts or steps.
That makes so much sense. But if you've got an AI writing 42,000 lines of code, how do you trust it? I mean, how do you ensure scientific rigor?
That is the most critical piece for science, right? Traceability. And Cosmos is built for it.
Oh, so?
Every single statement, every figure, every claim in a Cosmos report is cited directly back to its source. If it's a data finding, it links to the exact code in a Jupyter notebook. If it's a contextual claim, it links to the primary literature.
So you can follow its homework?
You can trace its entire line of reasoning, step by step.
But just because you can trace the logic doesn't mean the conclusion is right. We all know LLMs can make things up with great confidence. So what happens when you get humans to audit the claims?
Okay, so when they did that, independent scientists found that 79.4% of all statements were accurate.
So about 80% accurate, which at first sounds like, well, there's a 20% error rate.
Right, which seems a bit high, but when you consider the sheer volume and the complexity of the synthesis it's doing, getting 80% right on a first pass it's phenomenal.
Can we break that 79.4% down? I mean, where is it strongest and where is it weakest?
Sure, it's strongest on the pure data tasks. The data analysis statements were about 85.5% reproducible. Very reliable.
Okay, and the literature review.
Also very strong. About 82.1% of his claims from papers were validated. It's excellent at running code and summarizing what's in a paper.
So where's the weak link? Where does the human scientist still, earn their paycheck?
It's the interpretation and the synthesis. The parts where Cosmos tries to connect the dots and draw these big scientific conclusions.
How accurate was it there?
Only 57.9% accurate. This is the key limitation. It has a tendency to make overly strong claims, to misinterpret the nuance when it's connecting different pieces of evidence.
So the human's new job is basically filtering that 40% error rate in the big picture claims.
Exactly. The AI does the heavy lifting, the data crunching, the lit review, and the human provides that critical scientific taste and validation for the interpretation. The scientist is elevated, really.
They're no longer buried in code or PDFs. They're the critical evaluator.
Right.
So it's one thing to talk about accuracy, but the real proof is in replication. Let's get into the first case study, the one on metabolomics.
This was fascinating. Researchers gave it data from mouse brains undergoing hypothermia, cooling them down for neuroprotection. The goal was totally open-ended. Find the protective metabolic changes.
So just the raw data and the goal. No hints, no pre-selected papers.
None at all. And Cosmos almost immediately homed in on nucleotide metabolism as the most affected pathway.
And it found a specific pattern.
It did. It detected this inversion pattern. The precursor bases, the raw building blocks, were decreasing, while the final products, the phosphorylated nucleotides, were increasing.
And what does that inversion pattern actually tell us? What's the brain trying to do?
It's a sign of incredible biological efficiency. It means the brain is activating the nucleotide salvage pathway. Under stress from the cold, it's scavenging old materials and recycling them into new high-energy products.
So it's protecting itself without having to spend a ton of new energy.
Exactly. And the quantitative proof was just stunning. It perfectly reproduced the unpublished conclusion the human team had come to.
And the numbers themselves?
The log 10 fold change estimates were nearly indistinguishable, an R-squared value of 0.998.
For our listeners, an R-squared of 0.998 means the correlation is 99.8%. That's basically a perfect match.
It's an incredible validation not just of the finding, but of the method. It means you could use this to rigorously check human-led studies before they go to expensive trials.
And it wasn't just limited to biology. It also tackled materials science with those perovskite solar cells.
A really challenging field because the efficiency of those cells is so sensitive to tiny environmental changes during fabrication. Temperature, humidity, solvents, all of it.
So what did the AI pinpoint as the main bottleneck?
It confirmed what the scientists suspected, that thermal annealing humidity is the dominant factor. They called it the fatal filter.
The fatal filter.
Above about 60 grams per cubic meter of absolute humidity, the devices just failed. Consistently.
But here's the cool part, right? The bonus discovery, the thing the original researchers had missed in their own data.
Yes. While the human team was focused on the big variables, Cosmos's extended analysis found something new. It found a previously unreported linear decrease in the cell's short-circuit current density.
A linear decrease, so a direct, predictable relationship.
A very clear one. It correlated perfectly with the rising partial pressure of a solvent called DMF during one of the fabrication steps. And that discovery was later independently validated by the human researchers.
It just shows that the AI leaves no stone unturned. It finds relationships that human attention, which is naturally biased by hypotheses, might just skim over.
Exactly.
Okay, building on that, let's talk about the novel insights, where Cosmos isn't just rerunning numbers, but introducing whole new perspectives, like in genetics and myocardial fibrosis.
This was a very sophisticated study using Mendelian randomization, or NR.
So for our listeners, just briefly, what is MR?
It's a clever method that uses natural genetic variation as a kind of stand-in for a clinical trial. If a gene variant is linked to higher levels of a protein, and that same variant is linked to lower disease risk, you can infer a causal link.
Using our genes to establish cause and effect.
Precisely. The goal was to find proteins causally linked to myocardial fibrosis, the scarring of heart tissue.
And what did it find?
Both Cosmos and the human analysis independently identified superoxide dismutase 2, or SOD2, as the primary causal protein.
SOD2, that's a major antioxidant enzyme.
A critical one. The findings suggest that higher levels of SOD2 causally reduce that fibrosis, probably by neutralizing harmful free radicals that cause the scarring.
And I'm guessing the statistical agreement was, again, off the charts.
We're perfect. A correlation of 0.9991 for the shared proteins. But the real value add was the novel hypothesis Cosmos generated about how SOD2 was being regulated.
It went beyond just identifying the protein.
Much deeper. It proposed a specific mechanistic hypothesis. It suggested a genetic variant in the tail end of the SOD2 gene, the 3' UTR, was disrupting a binding site for something called a microRNA.
And disrupting that would do what?
It would change how the SOD2 protein is made, a post-transcriptional regulation mechanism. It's novel, it's biologically plausible, and it gives the human scientists a super-specific, actionable target to go and test in the lab.
That's incredible. It went from a broad correlation to a specific molecular mechanism.
Which brings us to my favorite part of this whole paper.
The Alzheimer's study.
Yes, where Kosmos didn't just follow a method, it effectively invented a new one for a really complex problem. Sequencing the events of a disease.
The question of what happens first.
The hardest question. So they had proteomics data from neurons in Alzheimer's cases, and these neurons were already computationally ordered along a disease timeline called pseudotime.
Can you explain pseudotime for us?
Think of it as a computational clock. You can't watch one cell get Alzheimer's for 20 years, so you take a snapshot of thousands of cells at different stages and computationally line them up from healthy to diseased.
Got it. A continuous timeline. So what unusual method did Kosmos propose for analyzing it?
Instead of just bidding cells into early or late stage, Kosmos proposed using segmented regression to find a quantitative break point along that continuous pseudotime axis.
Segmented regression. I know that from economics, for finding when a stock trend suddenly changes. Why is that so unusual in biology?
Because biologists tend to think in categories. This method treats the process as continuous and statistically finds the exact moment the underlying biology shifts. It's a much more precise tool.
And what did it find? What was the key breakpoint in the Alzheimer's timeline?
It found a highly significant breakpoint in the decline of the extracellular matrix, or ECM, which is the supportive scaffolding around the neuron. The breakpoint happened at 0.58 units of pseudotime.
So if the neuron starts showing tau pathology around 0.5 pseudotime, the ECM failure happens just after that.
Exactly. It suggests ECM failure is a downstream event. It follows the initial tau accumulation. That's a critical insight for knowing when and what to target with a drug.
And they checked its math.
They did. The human team used other methods, bootstrapping, sliding window correlation, and confirmed the exact same breakpoint. So Kosmos didn't just guess, it invented a statistically sound and correct method for the problem.
Which brings us to the final case study, the moment the AI discovers something the human scientists completely missed in their own data.
This was in a study of the entorhinal cortex in aging mice. The ENT is the first region hit by tau in humans, so it's incredibly important.
And Kosmos was just let loose on the data.
It was, and its unbiased approach found this coordinated systems collapse, involving two different cell types, the neurons and the microglia, the brain's immune cells.
A two-part mechanism that was overlooked. What was it?
Okay, part one. Kosmos found a systemic, age-dependent downregulation of P4 ATPase flippases in the vulnerable neurons.
Flippases? What do they do?
They're like molecular security guards for the cell membrane. They keep a molecule called phosphatidylserine, or PS, tucked safely on the inside of the cell.
And why is that important?
Because when PS is exposed on the outside of a cell, it's the classic eat me signal. It tells the immune system that this cell is damaged and needs to be cleared away.
Okay, so that's part one. The security guards are failing. What's part two?
Part two is that at the same time, the nearby microglia, the cleanup crew, showed an upregulation of PS-sensing genes. They were essentially getting primed to look for that eat me signal.
Wait, let me just get this straight. So the flippases, the security guards, are getting tired and letting the eat me signal flip to the outside of the neuron.
Right.
And at the exact same time, the local microglia are putting on their glasses and getting ready to spot that signal.
That's the perfect analogy. It's this coordinated, simultaneous event that provides a novel and very plausible mechanism for why these specific neurons are targeted for removal and aging.
And the human researchers who made the data missed this connection until Kosmos pointed it out.
It's the perfect example of AI augmentation. A human might focus on the biggest gene change and miss the subtle coordination between two different cell types. Kosmos, being systematic, didn't.
So, wrapping this all up, what does this all mean? Kosmos is obviously an incredibly powerful tool for unbiased, large-scale exploration.
It's accelerating months of human work into hours. It's changing the whole time budget of discovery.
But the scientist in the loop is still critical. Kosmos doesn't replace the human, it augments them.
Absolutely. You need that human oversight for the data quality, and most importantly, for filtering those synthesis claims, the ones that were only 57.9% accurate. You need human expertise for that scientific taste.
And let's not forget the current technical limits. It struggles with interpretation, can invent some weird metrics, and can't handle huge data sets over about 5 gigs or raw data-like images.
It still needs that well-formatted data from the scientists to get started. But for me, the most provocative part of this whole deep dive wasn't the speed.
It was the creativity.
It was the creativity. That moment in the Alzheimer's study. Cosmos, without being told, independently developed a new, statistically robust analytical method to answer a biological question.
So if Cosmos can invent new analytical methods that humans hadn't thought to apply in that context, what's next? What truly novel quantitative approaches could an AI like this develop that no human has even conceived of yet? And how quickly is that going to change the entire pace of research itself?